Good afternoon everyone. My name is Dr. Sukanya Mehra and I am from Department of Sciences, Khalsa College, Women. Today I am going to give you a lecture about plasma membrane. Before continuing with the topic of plasma membrane, let me give you a very small introduction about what is cell. Cell is a basic and a fundamental unit of life and it consists of all the cell organelles inside it. And to keep all the cell organelles intact, it contains a distinct organelle that is known as plasma membrane. Plasma membrane, earlier called as cell membrane and now it is called as plasma lemma, is a distinct organelle which forms a thin, transparent, elastic, regenerative and semi-permeable outermost covering which regulates the exchange of materials between the cytoplasm and extracellular environment to maintain homeostasis. Homeostasis is the process in which the regulation and the maintenance of ions, chemicals and compounds is done inside the cell as well outside the cell. Various structure models of plasma membrane have been given, but the model given by Singer and Nicholson in 1972, which is known as fluid mosaic model, is widely accepted all over. It is said to be formed of proteins as icebergs in the sea of lipids. Due to scattered nature of proteins, the plasma membrane is called mosaic. It is characterized by the following features. Plasma membrane is lipoproteinaceous and trilaminar. Lipoproteinaceous means that it consists of lipids as well as proteins, and trilaminar indicates the presence of 3 layers. Each protein layer is 20 Armstrong thick and phospholipid bilayer is 35 Armstrong thick. Phospholipid bilayer is fluid in nature, in which the phospholipid molecules show two types of movement. Transition movement, which is also known as lateral movement. Here, the molecules change their position within the same layer. The second movement is flip-flop movement. Here, the molecules of two layers can be interchanged. Proteins are globular and are of two types. First is extrinsic, which is also known as peripheral, and second one is intrinsic, which is also known as integral. In case of intrinsic or peripheral proteins, it lies outside the outer as well as inner surface of phospholipid bilayer. These are loosely attached by protein-protein interactions, so they are easily separable. Example includes spectrin that is present outside RBCs. RBCs are red blood cells. And another example is acetylcholine esterase that is found outside neuron. Second comes the intrinsic and integral protein. They are partially or completely embedded in the phospholipid bilayer. Their polar heads are projecting out of the phospholipid bilayer, while non-polar heads are embedded in it and held together by the ionic interaction, hydrophobic interactions and a specific interaction with the phospholipid molecules. These are strongly held to phospholipid molecules, thus they are not easily separable. Example includes rhodopsin, which is present on the retinal rod cells, and glycophorin that is present on mammalian RBCs. Some intrinsic proteins are so large size that they project on both the surface of phospholipid bilayer, and thus they are called as tunnel or transmembrane proteins. They act as passage for water-soluble materials along the concentration gradient. These occur either as single pass protein or multiple pass protein. Some of the membrane-bound proteins act as enzymes, certain act as receptors, and some act as carriers. Carriers are also called as permeases. These proteins provide structural as well as functional identity to the cell membrane. Each phospholipid molecule is a polar molecule and thus have specific ends. Hydrophilic head of glycerol and hydrophobic tail of two fatty acid chains. So, it is regarded as amphipathic in nature. Heads of phospholipid molecules of two layers are directed in opposite directions while tails of two layers face each other. This arrangement forms a water-resistant barrier which explains the rapid penetration of lipid molecules through cell membrane. In animal cell membrane, there is also sufficient amount of cholesterol which is responsible for bilayer fluidity of plasma membrane. Carbohydrates, mainly oligosaccharides. Oligosaccharides are the short chains of sugar molecules attached. They are also associated with the outer surface of plasma membrane. These either forms glycolipids or glycoproteins. These glycoproteins or glycolipids helps in cell-to-cell recognition and play important role in blood grouping, immune response, cancer and rejection of transplanted organs. These glycoproteins determine antigen specificity of the plasma membrane. This also makes the cell membrane asymmetric. These also forms a protective coat which is also known as glycocalyx. Due to this quasi-fluid state, biomembranes undergo dynamic changes. Example, renovation, extension, removal and folding. So, it shows variation in their form, size and functions. A biomembrane has two distinct surfaces, cytoplasmic and exocytoplasmic surface, because different types of phospholipids are found in two layers, different types of proteins in two protein monolayers. Glycoproteins and glycolipids are found only on the cytoplasmic side. Here comes the functions of plasma membrane. It helps in maintaining the steady state and homeostatic intracellular environment. A living cell must be an open system. So, it takes useful materials like water, oxygen, energy and nutrients which are essential for survival. This transportation includes two kinds of processes which is classified on the basis of energy consumption. First is passive and second one is active processes. Passive process includes simple diffusion and facilitated diffusion, whereas active process includes endocytosis and exocytosis. Further, active transport is of two types, endocytosis and exocytosis. Endocytosis is further divided into two categories, that is pinocytosis and phagocytosis. In case of passive transport, simple diffusion occurs. Here, the diffusion of particles can be solid, liquid or gas, transport from region of high concentration to the region of low concentration through semi-permeable membrane. The region of high concentration is also regarded as high pressure region. So, the definition can be molded as the simple diffusion is defined as the diffusion of transport of materials. Materials can be solid, liquid or gases from the region of high pressure to the region of low pressure through semi-permeable membrane. Here are certain factors that affect the rate of diffusion. Diffusion is directly dependent on speed as well as temperature, because they increases the kinetic energy, and kinetic energy further increases the diffusion potential of diffusion particles. However, the diffusion is inversely dependent on molecular weight of the diffusion particles. Osmosis is an example of diffusion in which the water molecules move from high concentration to low concentration through this membrane. Second comes the facilitated diffusion. Here also the particles move along the concentration gradient, but it involves the usage of carrier system or carrier protein. As it involves carrier protein, it is rapid as compared to simple diffusion. Next is active diffusion or active processes. Here, the energy expenditure is involved. So, the definition would be like, it includes the energy expenditure for the transportation of molecules from low region of concentration or pressure towards the high region of concentration or pressure through the semi-permeable membrane. So, here materials move against the concentration gradient. As it is energy dependent, it is highly rapid as compared to passive processes. Active processes includes endocytosis and exocytosis. The inward movement of particles is considered as endocytosis, however, the outward movement of particles is considered as exocytosis. Endocytosis is further divided into two categories, that is pinocytosis. Pinocytosis involves the intake of liquid molecules or the liquid nutrients, whereas phagocytosis involves the intake of large sized solid particles. Thus, in conclusion, we can say that cell membrane are not static but dynamic and helps in regulation of cell metabolism. Thank you.